Multi-compartment microfluidic neuron co-culture device fabrication begins with defining reservoirs on the PMMA block with a milling machine, followed by hot embossing of microchannel arrays that connect the compartments. Once a PMMA mold has been prepared, a PDMS mold master is replicated from the PMMA mold and coated with trichlorosilane. The final device is replicated from the PDMS mold master and assembled on a PDL-coated culture plate. Primary E16 to 18 rat neurons are then loaded into the soma compartment and cultured for 2 weeks, prior to adding P1 to 2 rat oligodendrocyte cells on top of axonal layers inside the axon-glia compartment for co-culture.
Hi, I'm Jaewon from Biosciences Laboratory in Electrical and Computer Engineering Department at Texas A&M University. Today we will show you a procedure for fabricating multi-compartment microfluidic neuron co-culture platform. We use this procedure in our lab to study central nervous system neuron-glia interaction in vitro. So let's get started.
To begin this procedure, use a micro-milling machine to make 3.5 mm deep compartments in a PMMA block. Define 1 soma compartment and 6 axon-glia.